Hi all. Today I'm discussing rapid papaya crop improvement through accelerated in vitro breeding and molecular diagnostics as we practice at Skyberry Farms. Skyberry Farms is located in the North Tropical Queensland in Australia. Skyberry is a diversified farm growing papaya, coffee, and other food crops. Skyberry red papaya is a market leader with over 70% of Australian market. Skyberry achieved excellence through innovation and best practices. Value addition is a tradition at Skyberry Farms, as evidenced by the variety of internationally acclaimed products like papaya vodka, coffee liquors, etc. that we market. Skyberry uses conventional and biotechnological approaches to improve its crops. Papaya is a superfood because it's rich in nutrients, vitamins, antioxidants, and is also low in sugar. In Australia, we grow red papaya, yellow papaya, as well as solo varieties. It may be interesting to notice that a red papaya has more potassium than a banana and more vitamin than an orange. Papaya is an excellent fruit. It's also a vegetable when it is green. It's also medicinal. Papaya is used for meat tenderizing, chill proofing of beer. The papaya seed oil can also be a raw material for biofuel production. Papaya production faces several challenges like requirement of good soil and climate. Genetic variability of seedlings is a problem with papaya like other crops. Unique sexuality of papaya with male, female, bisexual and hermaphrodite plants is another great challenge in making a large farm. There are many major pests and diseases on papaya. Papaya sticky disease caused by papaya melera virus, black leaf spot, phytophthora, fruit spotting bug, red spider mites are common problems with papaya farming in Australia. Papaya ring spot virus, which devastated the papaya industry in Hawaii, is fortunately not an issue in Australia. Why in vitro breeding? Conventional breeding faces challenges like requirement of huge amount of genetic variability to create useful hybrids. Unfortunately, papaya has low genetic variability. Papaya breeding takes 8 to 10 years to develop a new variety. Because of these challenges, Skyberry resorted into in vitro breeding, which can overcome most of the above challenges. At Skyberry, we use different in vitro techniques like cloning or micropropagation, somatic embryogenesis, soma clonal selection, cell line selection, induced variation and selection for our in vitro breeding. We achieved high efficiency cloning through nodal culture. A major challenge in high efficiency papaya cloning is achieving good routing. We achieved the octopus routing through intense research. Improvement in this particular step of achieving octopus routing reduced our difficulty at acclimatization and also helps the acclimated plants to be stronger in the field. Skyberry conducted large-scale field trial of cloned papaya, and the results indicated that cloning of papaya can counter sexuality issue by producing 100% hermaphrodite plants. Cloned plants are uniform, early bearing, and have low fruit set. They also have high vigor, thick stem, short internodes, and produce more number of fruits. Uniformity in growth and development also reduced management cost. We recorded 30% more yield and higher profit with the use of cloned papaya. The productivity of the papaya and the vigor of the papaya lasted even through the entire life of the plant. As you can see with this 18-month-old trial in the left, you can see the strong and vigorous, more productive tissue culture papaya compared to the seedling papaya on the right hand side. In 2016, PSD became a problem in Australian papaya farms. By 2017, it became a severe problem, and we were forced to find a solution to it to develop disease-free papaya lines to continue our farming profitably. Therefore, we developed high throughput somatic embryogenesis system. Cell culture was developed and cell line selection was performed using selection agents like acyclovir, ribavirin, and 2-thiouracil, the antiviral compounds. Plantlets were developed in large numbers through somatic embryogenesis. The plants developed through cell line selection were field tested after they were indexed for papaya sticky disease. In order to do efficient and rapid PSD diagnosis, we collaborated with Associate Professor Nitin Mantri at RMIT University in Melbourne and developed an RT-PCR method that was very efficient and rapid in detecting PSD, even in the callus cultures and also cultures growing, shoot cultures growing in the laboratory. We further developed lateral flow device method for detection of PSD in the nursery stock. This method can also be applied to detect PSD in the field plants. Further, we developed mutation breeding in order to achieve disease resistance. So we treated the embryogenic callus with gamma rays at 10 to 100 K concentration, regenerated several plants through somatic embryogenesis, disease indexed for PSD, cloned PSD free lines, and field tested. At the laboratory stage, we could identify several kinds of obvious mutants like albino, spindly, curly leaf, dwarf, etc. Normal looking mutants were field tested, and when they were tested for PSD, when they were indexed for PSD, we found that at 70 gamma radiation actually nearly eliminated PSD from the callus cultures. And there was very few regenerants that showed PSD presence after 70 Gauge radiation. So the normal looking mutants were multiplied and field trialed. Out of that, we got some excellent papaya lines. The most productive plant that produced more than 400 fruits in 18 months with a Brix of 14 to 15 and sweet red flesh is named as Skyberry 35 to commemorate the 35 years of Skyberry papaya farming at this North Queensland location. In conclusion, cloning alone improved productivity of papaya by 30% or more. Average yield with seedling was 64 tons per hectare. We enhanced average yield to 85 tons per hectare with the clones, about 33% productivity gain in 5 years' time. Cell line selection generated improved PSD-free papaya lines. Mutation breeding, aided by molecular diagnosis, further increased productivity. We thankfully acknowledge the Australian R&D tax incentive scheme and the Innovation Connection grant provided by the national government that helps us to achieve these milestones in such a short time. This graph shows the tremendous increase, 2% increase in the sugar content of Skyberry papaya in 7 years' time through this in vitro breeding research. Thank you all for patiently listening. Any questions, please contact me. Thank you.